Namaskar. Very warm welcome to all the viewers and learners in this live interaction of CIET and NCERT. I am Renu Bhatt with you all and you are watching us live on PM eVidya channels from channel number 6 to 12. Apart from all these channels, you are with us on our YouTube channels as well, that is NCERT Official and NCERT PM eVidya class number 6 to 12. And dear learner, this is our special session on virtual lab, and the topic that we are going to discuss today in this specific session is to study the phenomena of osmosis. And before we proceed further, let me tell you something about virtual lab in a brief. And as the name itself suggests, it's a laboratory which is virtual, and any student from anywhere with the help of internet can access and perform their experiments. Actually, virtual labs provide remote access to simulation based labs in various subjects, and it offers various resources like videos, simulation, animation and self-assessment. And the best part of virtual lab is it enables the student to learn at their own pace. No, it's not the alternative of physical lab, but with the help of virtual lab, any student can enhance their knowledge and confidence before conducting any experiments. So let's quickly start this session and let's meet our expert, and she will let you know about the other benefits and positive points of virtual labs and the simulation as well. So let's quickly meet her. You are Dr. UPASANA Sharma. You are academic consultant from CIET NCERT New Delhi. Very warm welcome, ma'am. स्वागत है आपका.
Thank you.
And before we proceed further, let me give you some certain information to all the students and teachers out there in case they have any query or they want to know more about virtual lab or particular this topic. You can reach out to us through our various mediums. We want you to call us on our telephone number, that is 8800 440 559. Either you can drop a mail as well with all your queries and feedbacks. Our email address is diksha.training@ciet.nrc.in. And if you have joined us through our live streaming, then you have to go to the live chat box and then only you can drop your comment out there. Our expert will be more than happy to answer all your queries and doubts if you have any. So let's quickly start this session. And you must be wondering about if virtual labs are that much good, ma'am. So where they can find virtual lab? What is the navigation? Could you please explain to all the viewers, ma'am?
Yes, definitely. First of all, to access the virtual lab, go on any web browser and type diksha.gov.in. You will land on this kind of a page. When you scroll down a bit, you can see the focus areas of Diksha are given. Scroll down to either right or left. You will find the tab of virtual lab. Click on that and then you will redirect it to this virtual lab home page. In the starting, you can see about virtual lab is mentioned, where the NEP envisions regarding the virtual lab is mentioned. When you scroll down a bit, you can see the content is divided according to the classes. This is from class 6th to class 12th. Click on any class, click on the explore button. You will see that the content is in English and Hindi medium. Click on the medium, you can see that the content is according to the subject. This is the table of content page of virtual lab. Here on the right side, you can see that the table of content or the experiments are mentioned. These experiments are based on the NCERT textbook. Today for this session, I will take one experiment from the class 9 chapter 5, that is the fundamental unit of life. Click on the experiment. When you click on the experiment, then you can see the various resources are given. These resources are the theoretical aspect of the experiment, the procedure of the experiment, the animation, the video part, the other resources for further studies, the simulation and the self-evaluation. Today we will try to understand what actually is the process of osmosis. So the objective of the experiment is to study the phenomena of osmosis. Now what actually is osmosis? Osmosis is a process of transfer of water molecule from a region of higher water concentration to the region of lower water concentration, and also with the region of lower solute to the region of higher solute. This takes place with a selectively permeable membrane. This process continues until and unless the concentration of both sides of the membrane becomes equal. Now, what are the some characteristics of osmosis? See, the process of osmosis takes place from a region of a lower region of lower solute concentration to the region of higher solute concentration. This takes place with a membrane. Now what exactly is this membrane? This membrane is semi-permeable in nature, or you can say it's a selectively permeable. Now what exactly is selectively permeable? By means of selectively permeable, it means that it allows the passage or the movement of selective molecules, or it allows the movement of water molecules, and also it restrains some other molecules. And what actually it restrains? It restrains some solute molecules like such as ions or larger particles. Now the second important characteristic is the concentration gradient. See, the process of osmosis takes place in a concentration gradient, as I have just mentioned, that the movement takes place from a lower solute concentration to a region of higher solute concentration. This is driven by the natural tendency of solvent molecule to move from the region of higher water concentration to the region of lower water concentration, and this process takes place to the limit where the process of equilibrium has been achieved. Now here I want to point out one thing, that here by equilibrium it does not mean the concentration of solute on both sides become equal. In general, it refers to the equal concentration of the water. Now as you can see from the diagram, that the movement takes place from the lower concentration solution to the higher concentration solution through a semi-permeable membrane, that is a selectively permeable membrane, and the movement of solvent from low to high solute concentration until unless where the equilibrium has been achieved, or you can say the same concentration has been achieved on both sides of the membrane. Now when we are talking about osmosis, we are talking about solutions. So basically there are three types of solution. Number one is hypertonic solution, number two is hypotonic solution, and the third one is isotonic solution. Now what exactly is a hypertonic solution? A hypertonic solution has a higher concentration of solute. And what exactly happen in a hypertonic solution? In a hypertonic solution, when you placed any cell, the water tends to move out from the cell, causing the cell to shrink, or you can say it undergo the process of plasmolysis. This is because the water moves from a region of lower solute concentration to an area of higher solute concentration. Now the second is hypotonic solution. The second example of hypotonic solution, I can say, when you place any resin under water, when you put resins under water or soak resins overnight, then you can see that it swells up in. Then why itself swells up in? It is because the hypotonic solution. A hypotonic solution has a lower concentration of solute compared to another solution, which makes the solution to move into the cell, causing it to swell or even burst. This is also called as cytolysis. And the third is the isotonic solution. An isotonic solution has the same concentration on the both side of the membrane, and there is no effect on the size of the cell. When you keep anything in an isotonic solution, generally it has been found that there is no change in the size, it neither shrink or swell. The isotonic solution generally used in the medical settings like for IV fluids to prevent cells from shrinking or swelling. Now as I have just discussed about the semi-permeable membrane, this semi-permeable membrane allow or it permits the passage of any certain molecules or ion that depends upon the size, the charge, the solubility, and it restricts the depending upon the size and the solubility of the molecule. Now, what exactly is deplasmolysis? Deplasmolysis is a process by which a plasmolysed cell, or you can say a cell which has lost its water, is restored to its original turgid state. Now, what is actually a turgidity? Turgidity refers to the state of being swelling up. Now, what is the importance of osmosis? We are talking about the osmosis and how it is important and how it is useful in our daily life. Osmosis helps in the transport of nutrients. It maintains the turgidity of the cell. The cell membrane of both plants and animals form a selective barrier between the cell and the environment. Thus, it helps in refraining the animal cell or the plant cell from outer or environment, the substances which are toxic in nature, it refrains to come into the cell. This plant and their parts move in a regulated manner due to the induction of cell turgor. The selectively permeable membrane allows the cell to regulate the flow of necessary substances into and outside of the cell. The osmosis controls the opening and the closing of stomata, and it is one of the reasons to raise the why the water raised to such height into a tree. Now, the types of osmosis. Depending upon the solution, the osmosis can happen is of two types, end osmosis and exosmosis. The process of end osmosis takes place when the water comes into the cell. We call this end osmosis, and in result what actually happens? This cell becomes swollen up. And the other one is exosmosis. In exosmosis, a substance found in a hypertonic solution causing the cell to become flaccid or solvent molecule leaves the cell. In short, we can say it makes the cell to shrinken up. Now, what exactly a learner will understand at the end of this experiment or process? A learner will understand the process of osmosis, the difference between the end osmosis and exosmosis. The students will be able to do more such type of experiments in real lab settings, or the learner can also assess the animated and the other resources and can learn from that as well. So, this is all about the theoretical aspect of process of osmosis. As you can see, when you scroll down a bit, the procedure is given. In the procedure, the materials required for this experiment is mentioned, like what exactly a learner will require. A learner required the 2 raw eggs, the learner required a dilute HCl, a salt or a sugary solution, beakers, etc. And the procedure is mentioned for the real lab setting as well as for the simulated environment. So, a learner or a teacher can use this pre-lab manual in case of the absence of the manual present in any real world or the physical laboratory. Before performing the simulated experiment, learner can follow these steps like how to drag and drop any particular thing. And at the end, the learner can observe and make analysis, thus why this particular phenomena, why this particular process and how this particular process is taken place. The learner can also watch the animated videos. These animated videos are very engaging. So, a learner can go through these animated videos and learn the steps of the process of osmosis. And learner can even watch videos. So, in short, a learner can go through all these resources, can read out about the experiment, can read out related to the procedure required for doing any particular experiment, and also for further studies can go through to the website or the inputs mentioned in the resources. Now let us do the experiment. Click on the simulation. So this is the interface of the experiment. As you can see on the left side, instructions are given, and on the extreme right and on the top, the 3 tabs are given. The first one is reset, another one is help, and the third one is minimize. A learner can minimize or maximize the experiment according to its need. A learner can take help. If the learner gets stuck anywhere, can reset or restart the experiment from anywhere. Now let us start the experiment. Suppose I do not know what step I need to follow. So I can take the help from the help tab. So it is asking me to drag and drop the eggs into any of the solution. This solution is of hydrochloric acid. I am dragging and dropping the first egg into the beaker. The beaker contains the dilute hydrochloric acid, and I am dragging and dropping the another egg into the another beaker which also contains the dilute hydrochloric acid. See, as you can see, this experiment requires a learner to wait for certain hours, and in real world setting, in a real classroom setting, it is not possible for a teacher to wait this much time to perform any experiment or to observe the analysis or the result of the experiment. So after 4 hours, what happened? As you can see, the eggs become de-shelled. These eggs are made up of calcium carbonate and it gets dissolved in the dilute hydrochloric acid. Now it is asking me to drag and drop one of the egg into the beaker containing the water, and the another egg also in the beaker containing the water. Now what happens next? See, you can also reset this experiment anytime. Suppose you get stuck anywhere, you can reset this experiment anytime. See what happened after the 4 hours. As you can see, there are 2 type of solutions are given. One is the water solution and the other one is the salt solution. Now I need to drag and drop one of the egg into the water solution and the another one in the salt solution. As you can see, there is difference between these 2 type of solution. One of the solution is the hypertonic solution. A hypertonic solution is the solution where the concentration of solute is higher, and one of the solution is simple water, that is the hypotonic solution. As you can observe, there is certain changes in these 2 different eggs. As you can see, one of the egg has swollen up and another one has shrunken up. Now why this happens? In one, the solution, the egg which has kept into the water has swollen up. It, the de-shelled egg when placed in water gets swollen up because this solution is, or this solution contains the hypotonic solution. The water enters from the semi-permeable membrane, and in the another solution you can see, that is in the salt solution, you can see the egg has shrunken up. Why this happened? Because this solution is hypertonic solution. Here the concentration of the solute is higher. Here the process of osmosis has taken place in the first beaker, and the process of osmosis has taken place in the another beaker. Now from the inference you can understand that in the process of osmosis the water gets inside the cell, and in the process of osmosis the water gets outside from the cell, making it to shrinking up. And this depends upon the concentration of the solute, whether the solute is hypertonic or the solute is hypotonic. So in this way a learner can easily understand the process of osmosis. A learner can easily differentiate between the different type of solution, whether the solution is hypertonic solution or hypotonic or the isotonic solution, and how the process of osmosis has taken place and how the process of osmosis takes place. A learner can do this experiment as many times a learner want, and if a learner thinks that he or she is now perfect enough, then he or she can go through the self evaluation tool. This self evaluation tool is also very useful for the teacher. A teacher can use this tool in the classroom to evaluate various types of learner in the classroom, or even to analyze the understanding of a single learner. There is various type of questions are given. A learner can go through a learner can go through all the type of question, can understand now whether he or she can understand the topic completely. And if he thinks that he or she needs to redo the experiment, can redo the experiment anytime, or can go through the animated videos, can go through the procedure, can go through the theoretical aspect as well. So this is all about the process of osmosis, how a learner can perform this experiment in real world setting as well as in the virtual world setting, and what a learner or what steps a learner need to perform in the real world, and how a real world experiment is slightly different from the virtual world, or what all benefit a learner can get by performing this experiment into the simulated environment. As I have just said that these type of experiment or these type of procedures which are given online, this provides a add on to a learner, to add on to its knowledge, to an add on to its interest. It also motivates the learner. It also makes this classroom process a little bit engaging and fun loving for a learner.
Definitely, ma'am. So it means virtual lab, learning from virtual lab is quite engaging for both, I mean, teachers as well as for the students, right?
Yes, yes.
Help is always available and any student can perform n number of experiments, right? It's quite interesting. But it's time for us to wrap up this session here only. Before that, let me thank our expert. Thank you so very much, Upasana ma'am, for being with us. Thank you, ma'am, for this insightful session. Thank you so very much, dear viewers, as well. And before I wrap up this session, let me share all about the other sessions detail as well. At 12 p.m. we will be back with our another live interaction for mathematics 9th standard students. The topic would be surface areas and volumes. At 2 p.m. we will be back with live interaction for 1st standard mathematics students. The topic is fun with numbers part 1st. At 2:30 p.m. English session is there for 1st standard students, fun with pictures, followed by Urdu 2nd standard students. Topic would be Barsat at 3 p.m. At 3:30 p.m. Hindi 2nd standard students will be having their discussion on Tilluji. At 4 p.m. this is our time for online training program. The topic is cyber अपराध की अवधारणा और हाल के रुझान. At 5 p.m. Sahyog session will be there for adolescent students for their mental well-being. And before I wrap up, let me tell you about the topic for tomorrow's virtual lab session, and the topic is to identify washing soda or baking soda among given samples of chemicals, and this session would be for chemistry 10th standard students. So Me, Renu Bhatt, is taking your leave, but you stay tuned to PM eVidya channel for more informative programs. Namaskar.